This video goes along with chapter 13, which is meiosis. We'll start with the inheritance of genes. Parents give their offspring coded information in hereditary units called genes. A gene is a specific nucleotide sequence that's found within a chromosome. In plants and animals, reproductive cells called gametes meaning sperm or egg, transmit genes from one generation to the next. DNA in eukaryotic cells are packaged into chromosomes in the nucleus. Each organism will have a characteristic number of chromosomes. For example, humans have a total of 46. When we talk about somatic cells, we are referring to all the cells that are not sperm or egg and have the full set of chromosomes. So for humans, they would have the full 46. Each chromosome is a long DNA molecule that contains hundreds to thousands of genes. And a gene's location on a chromosome is called a locus, or the plural form of that is loci. So that's kind of the need to know before we move into what we're getting into next. First, we'll compare asexual and sexual reproduction before we get into the details of sexual reproduction. During asexual reproduction, a single individual is the sole parent and passes copies of all of its genes to the offspring. So there's not going to be a fusion of gametes at any time. Some example organisms that do this process are both yeast and amoeba. Individuals that reproduce asexually give rise to clones because they're all genetically identical individuals. During sexual reproduction, two parents give rise to offspring, which gives us a unique combination of genes every single time, and that will vary among offspring even of the same parents, except for the case of identical twins. But if you have brothers or sisters or know someone who does, you'll know that they look similar, but also different, and that's because we have a different gene combination every time, unless you or someone you know is an identical twin. Fertilization and meiosis alternate in sexual life cycles. So when we talk about a life cycle, we're talking about the generation to generation sequence of stages in the reproductive history of an organism. And we will look at humans as our example organism here. As I said, human somatic cells have 46 chromosomes, and so they'll have 2 chromosomes each of 23 different types. And we can use a karyotype, which is a display of chromosomes, to visualize them, and they're matched by size. 2 chromosomes of the same type will have the same genes, even if they are different versions of that gene, and they are called homologous chromosomes or homologs. The 23rd pair are the sex-determining chromosomes. So for females, that would be XX, and for males, that is XY. And we call pairs 1 through 22 autosomes, and pair 23 the sex chromosomes. So if you look at the karyotype here, I can see 23 pairs of chromosomes, and we number them based on size. So chromosome 1 is the largest, 22 is the smallest, except for, the Y chromosome. So this individual's karyotype, I can see that it is a male because there's 1 X and 1 Y chromosome and 2 of everything else. We'll look more at karyotypes in a bit when we get to mutations. Let's look at the sets of chromosomes in human cells. We inherit 23 chromosomes from each parent to give us the magic 46. And in bio, we are going to use small n to represent a single set of chromosomes. A diploid cell contains 2 sets of chromosomes, so that will be all somatic cells. But gametes, like sperm or egg, will only contain 1 set of chromosomes, and we call them haploid cells. Each species has a characteristic haploid and diploid number. As we all know, for humans, that's 23 and 46. But for a fruit fly, it is 39 and 78. The number doesn't necessarily mean anything about the complexity of the organism. For example, little tiny fruit flies might have more chromosomes than we do. That just tells you how many linear pieces there are of DNA in the genome. So my guess would be that maybe fruit flies are a little bit smaller. If you look at like sheer numbers of nucleotides, it's probably a little higher here. The human life cycle begins when a sperm from father fuses with an egg from mother in a process called fertilization. This newly fertilized egg is called a zygote and is the very first diploid cell. It's kind of weird to think that you used to be just one tiny diploid cell, and that one cell undergoes mitosis a ton, even within like the first 24 hours after fertilization. And that one zygote eventually turns into a sexually mature adult that can produce sperm or eggs and therefore a new offspring. So that's kind of the life cycle of humans, which you can see here in picture form. The only cells not produced by mitosis are gametes, and gametes will develop from specialized cells called germ cells in the gonads, which are ovaries or testes. And gametes are formed during meiosis, which reduces the number of chromosomes by half and produces haploid cells, i.e., sperm or egg. There are a variety of sexual life cycles. With humans and most other animals, gametes are the only haploid cell, and that's the same process that we have recently looked at. In plants and some algae, they have alternation of generations, which includes diploid and haploid stages that are multicellular. In most fungi and some protists, after gametes fuse and form a diploid zygote, meiosis occurs without a multicellular diploid offspring developing. And so here, meiosis produces haploid cells that divide by mitosis and give rise to unicellular descendants or a haploid multicellular adult. As a note, haploid and diploid cells can divide by mitosis depending on the type of life cycle, but only diploid cells can go through meiosis. Otherwise, there would not be enough chromosomes, and that's an issue. So here is that in picture form. You see with plants, you can see the alternation of generations, and then you can see the life cycle of the fungi and protists there. Meiosis reduces the number of chromosome sets from diploid to haploid. You'll notice that the steps of meiosis resemble the corresponding steps of mitosis. DNA replication happens before meiosis as well, but there are two cell divisions that we call meiosis 1 and meiosis 2. The end result is 4 daughter cells with half as many chromosomes as the parent cell. So it makes 4 haploid cells every single time. So here is a general overview of meiosis. So during interphase, we have our pair of homologs there, and then we have a little bit of DNA replication, so we now have a copy of everything. And then during meiosis 1, our homologous chromosomes separate one to each cell, and then both of those cells go through a second division, or meiosis 2, where those chromatids, which are each piece of that X shape, get separated to form 4 cells that are haploid, because we started with 2 and now each cell has 1. Starting with meiosis 1, the first step of meiosis 1 is called prophase 1. And this is perhaps the most complicated and biggest event of the whole cycle. And it's going to be different from prophase 2 when we get there in a minute. So what's going to happen is that our duplicated homologous chromosomes, here red is representing maternal and blue is representing paternal, are going to pair up to form a tetrad, and they go through a process called crossing over that we'll look at here in a bit. And so what's going to happen during crossing over is they're going to exchange little bits of pieces so that an individual chromosome is going to contain a combination of maternal and paternal genes instead of it being separate. And so, but then like all the normal prophase stuff happens too, where before crossing over, the chromosomes condense, the nucleus disappears, blah, blah, blah. So that picture there is kind of a late prophase event because you can see crossing over happening at a couple different places. And then during metaphase 1, the pairs of homologous chromosomes are going to line up in the center. the difference between this and mitosis is that they're lining up in pairs and not, 46 all in a row, for example. And then during anaphase 1, the homologous chromosomes of each pair are going to be pulled apart. And then telophase 1 and cytokinesis happens, where we have our nuclei reforming around the genetic material, and then we have our pinching cleavage furrow to form two cells. So that is meiosis 1. During meiosis 2, during prophase 2, same kind of things happen, but there is no second crossing over event. So we're starting with the two cells that are straight out of meiosis 1, and the same things are going to happen. The nucleus disappears. We have spindle fiber formation and attachment and all that other stuff we know from mitosis. And then during metaphase 2, the chromosomes are going to line up single file this time. Anaphase 2, the chromatids are pulled apart. And then telophase 2 and cytokinesis happens, where we have the nuclei reforming around the genetic material and cytokinesis. And so with crossing over and all that, you can maybe kind of see from this picture, but each cell looks seriously different. It's not, two red ones and two blue ones. There are segments that are on one chromosome that are partially red and partially blue, and that's from crossing over, which we will look at here. So this is after interphase and like later on in prophase 1. The chromosomes have already been duplicated. Sister chromatids are held together by cohesions. Each pair of homologs associate along their length, so they line up together. And the DNA molecules of two non-sister chromatids are broken at precisely corresponding points, and the chromatin of the chromosomes start to condense. And then a zipper-like protein complex begins to form, attaching one homolog to the other, and the chromatin is still condensing. And it's that condensing that makes it visible to us with a microscope. And then once the complex is fully formed, the two homologs are in synapsis. And during synapsis, the DNA breaks are closed up when each broken end is joined to the corresponding segment of the non-sister chromatid, producing crossing over. And then they get out of that phase, and then they're ready to go into metaphase 1 at that point. An important point here is that crossing over is not going to occur with sister chromatids because they are identical. So it does have to be the non-sister chromatid for crossing over to occur. Let's compare mitosis and meiosis. I know I've done a little bit of that here. So in our example, the parent cell, so what we're starting with before we go through either mitosis or meiosis, has a diploid number or 2n number of 6. And so some things they have in common is before any of this happens, we have replication of the chromosomes before prophase starts. One difference here is that in meiosis 1, like we said, during prophase 1, we have crossing over. That does not occur with mitosis at all. Another difference is that during metaphase of mitosis, the individual chromosomes are lining up in the middle, but in metaphase 1 of meiosis 1, they're lining up with their homologous buddy. And then obviously the end result is going to be a little bit different. With mitosis, we have two identical daughter cells that are both identical to each other and to the parent cell. But with meiosis, we have 4 genetically different haploid cells. And we've already been through all that, but that is there for your review on your own. There are 3 mechanisms in sexual reproduction that lead to most of the variation among offspring. And we'll look at each of these in sequence: independent assortment of chromosomes, crossing over, and random fertilization. So, starting with independent assortment. There is a random orientation of pairs of homologous chromosomes during metaphase 1. So there is a 50% chance the maternal chromosome is closer to the center and a 50% chance that the parental chromosome is closer to the center. These chromosomes are going to assort randomly, which is why it's called, well, never mind, we'll get there later. Something to note here is that these chromosomes will assort independently of one another. So just because in possibility one, like just because the maternal chromosome is on the right hand side on the top, that does not mean that the next set will also align that way. It has a 50% chance of being the other way. So this idea is called independent assortment. And there are 2 to the n possible combinations where n is the haploid number. So for humans, when there are 2 to the 23rd combinations of how our homologous chromosomes could align, which is about 8.4 million. Crossing over also leads to genetic variation because it produces recombinant chromosomes, meaning it has genes from both parents. Humans have about 1 to 3 crossover events that occur per chromosome, and that that's going to depend on the size. So even if that's, let me just think about, even if that's 2, that's quite a few crossing over events, and we've already been through that process. But there's a maybe easier diagram for you to see in Figure 13.2. And finally, random fertilization. So we'll throw some numbers out there because this is what we know. Each male and female gamete has about 8.4 million, that's our 2 to the 23rd, possible chromosome combinations from independent assortment. Therefore, when we have fertilization or fusion of a male gamete with a female gamete, that zygote has any of 70 trillion diploid combinations because we're multiplying like the probability of this and this. And so that's how many combinations there are, but that is not including variation due to crossing over, which we also know is high. So it's really even higher than that 70 trillion. So those 3 events are truly what make you,